A microscopic, jagged fragment navigates the slipstream of a human blood vessel. This fragment is known as a microplastic. These tiny synthetic particles shear off of food packaging, clothing, and industrial waste, entering our drinking water, our food, and the air. This diagram shows a cross-section of a human artery with plaque buildup. Zooming into the dense lipid matrix reveals distinct, jagged plastic geometries lodged directly inside the biological tissue. Clinical observation confirms that patients with higher concentrations of these embedded plastics suffer from significantly worse cardiovascular outcomes. We must determine whether these microplastics are inert debris caught in the wreckage or the active drivers of the arterial damage itself. Standard cardiology models attribute plaque formation to high cholesterol, poor diet, and metabolic dysfunction. A study from the University of California, Riverside directly challenges this established framework. Researchers designed an experiment to isolate microplastics from traditional risk factors, observing how the particles affect the vascular system on their own. Conducted with Harvard Medical School and Boston Children's Hospital, the study set out to provide definitive answers on the physical toll of plastic exposure. The findings provide compelling evidence that microplastics operate as direct cardiovascular pathogens. To test this, researchers used the LDLR deficient mouse. This biological model lacks the receptor needed to clear bad cholesterol from the blood, making it the standard baseline for studying human heart disease. This matrix visualizes the experimental design, separating mice into male and female cohorts. All subjects were restricted to a strict low-fat, low-cholesterol diet. The primary variable was a daily dose of microplastics for exactly 9 weeks. This dosage was carefully calibrated. Rather than inflating the numbers, the team used volumes that accurately reflect real-world human exposure levels. By maintaining a healthy diet and using realistic plastic concentrations, the researchers neutralized the traditional culprits of heart disease. In this controlled environment, any severe arterial plaque development could be traced solely to the microplastics. At the end of the 9-week exposure, researchers checked the subjects for traditional cardiovascular warning signs. This dashboard tracks the metabolic health of the subjects. The first graph maps body weight. The subjects experienced no abnormal weight gain. Looking at systemic cholesterol, the levels remained completely stagnant. Comprehensive lipid profiles showed no negative changes whatsoever. Microplastics initiate severe vascular injury while bypassing every standard metabolic indicator used in clinical diagnosis. While the metabolic blood work appeared normal, a physical examination of the arteries revealed a catastrophic breakdown. This graph highlights a binary split between how the male and female cardiovascular systems reacted. After 9 weeks of exposure, the female mice showed no significant increase in plaque formation. They maintained the healthy baseline. The male data shows a sudden acceleration in atherosclerosis. The pathology in the male subjects included a 63% increase in plaque, occluding the aortic root. Even more severely, researchers measured a 624% increase in plaque within the brachiocephalic artery, the vessel supplying blood to the head and right arm. This localized explosion of plaque proves that in male subjects, microplastics rapidly accelerate arterial blockages, operating independently of cholesterol-driven models. How does an inert piece of plastic cause a 624% localized explosion in arterial plaque? The answer lies within the endothelial cells. These form the single-cell layer lining the interior surface of the blood vessels. These tiny, jagged particles physically wedge themselves deep into the delicate endothelial lining, completely warping the surrounding cellular structure. This physical intrusion immediately triggers an aggressive inflammatory response from the surrounding biological tissue. The presence of the synthetic material forces the gene activity of the endothelial cells to shift, promoting the growth of plaque. The resulting arterial blockage is essentially an emergency layer of inflammatory scar tissue forming over the embedded plastic shrapnel. The data raises a difficult question. Why did the male cardiovascular system suffer catastrophic damage while the females remained statistically immune? Researchers have not yet isolated the definitive biological mechanism, but they are investigating several hypotheses. Leading theories focus on differences in sex chromosomes, hormonal variances, and the cardiovascular protective effects of estrogen. We must also ask how closely this mouse model maps to human biology. Parallel laboratory tests directly exposed human endothelial cells to microplastics. The cells exhibited the exact same inflammatory and genetic responses observed in the mice. This suggests the human vascular system is vulnerable to these direct plastic-induced injuries. If our arteries are currently embedding these inflammatory particles, what is the clinical procedure to remove them? At this time, there is absolutely no known medical method to extract microplastics once they enter human tissue. Because extraction is currently impossible, the only evidence-based defense against this form of heart disease is aggressive exposure reduction. Scientists recommend eliminating single-use plastics, using glass or stainless steel containers, reducing intake of highly processed foods, and filtering indoor air. Heart disease is evolving into an environmental concern driven by the synthetic dust we ingest every day.